Hi, I'm Carl Petersen. I'm a professor of neuroscience at the École Polytechnique Fédérale de Lausanne. This is what it looks like, and we're located in Switzerland, where we have lots of beautiful mountains. So when the Berlin Wall fell on the 9th of November 1989, I was just a first-year student of medicine at the University of Oxford. And when we heard the news, we went down to the pub and celebrated. As a child, I was always fascinated in science and technology, and my highest wish was to become a physicist, a researcher in the field of the basic mechanics of how the world works. As it turns out, I've become a neuroscientist, but I call myself also a biophysicist. So in part, I think my dream has come true. I'm a neuroscientist. My work addresses two important challenges for mankind. The first is the intellectual challenge. How does the brain work? The second is the challenge of how we can develop therapies to treat the many devastating brain disorders. We address these challenges by developing new technologies to measure, manipulate, and to model brain function. And through the combination of these new technologies, we want to understand how neural circuits give rise to brain function in health, how the circuits become altered in disease, giving rise to dysfunction, and how we can change those circuits in order to cure the many brain diseases. Reward-based learning drives important aspects of animal behavior. We all do things in order to obtain rewards. And the way that we know what to do in a given context, what our behavior should be, is through our experience. We learn that in a given context we should do something, and if we do that, we get rewarded. And that reinforces that context-specific behavior. We want to understand the detailed mechanisms by which the brain learns what to do in a given context when a given sensory stimulus is delivered. And so we make these investigations in mice to get a quantitative understanding of the underlying neuronal circuits. In our experiments, we deliver a very simple stimulus, a single deflection of a single hair on the snout of a mouse. Through a process of trial and error learning, the mouse learns that if it licks a reward spout immediately after the sensory stimulus, then it's delivered with some sweet sugar water, its reward. And through trial and error learning, mice can learn to accomplish the simple goal-directed sensory motor transformation, taking a whisker stimulus into licking and accompanied by reward. So the essential question for us in our research is how is sensory information, the whisker deflection, transformed into motor output, licking, through reward-based learning? What changes in the brain in order to link sensory and motor together with rewards? We investigated this through multiple different ways. We measured activity in the neural brain through electrophysiology and optical imaging, where we can track the changes in highly specific neurons and neural circuits across learning. We can manipulate the neuronal circuits and individual cells in the brain through optogenetics, where we can shine light to stimulate neurons or inhibit neurons. And for example, we can optogenetically program learning behaviors. And we can also computationally model the activity in the brain to make sure that we have a quantitative understanding of how different neurons talk to each other and what changes occur that drive learning. And in part, we do that together with the Blue Brain project at EPFL. What we've discovered is a highly specific circuit linking sensory and motor parts of the mouse brain, and those circuits get linked up through reward-based learning. The whisker deflection is initially processed in sensory cortex, primary sensory cortex, S1. That information is passed on to secondary somatosensory cortex, S2, which in turn informs secondary motor cortex, M2, and that drives activity in primary motor cortex, M1, that initiates the licking in order to obtain a reward. So we think we have one of the most detailed and most quantitative understandings of how reward-based trial and error learning changes neuronal circuits in the brain, transforming sensory information into motor output. With over 150 million people in Europe alone suffering from various different mental health issues, understanding the mechanisms of brain function is of high importance. Basic science contributes neural circuits that provides a basis for designing new therapies for the many devastating brain disorders. My work has direct implications into schizophrenia, which has perceptual issues, depression with motivational issues, and Parkinson's disease, where there are deficits in the initiation of movements. By investigating mouse models of these diseases, we will come up with neural circuits and targets for new therapies that will have a major impact upon society in the future. Neuroscience is a very young field and much remains unknown. Until we have a quantitative model that replicates the neuronal activity and the behavior of an animal, we don't know whether we've fully understood what's going on and what underlies a behavior. And for me, what keeps me awake at night is the concern that we may not be able to get sufficiently good models, sufficiently good data in order to have complete explanations. One of my best friends is a teacher and is interested in another aspect of our research, and that's how we can improve learning outcomes of students, how we can speed up learning, and that's of course something that we're also interested in for the mouse. How can we improve the learning of the mouse? So there are many interesting impacts of our research.